surprised to see that absolutely there was no growth or development of the nervous system. It was not improper, it was utterly not formed. So, by these experiments, he concluded that mesodermal cells, they stimulate the cells of the ectoderm, which in turn produces the nervous system. So, this phenomena is also known as embryonic induction, and now we will see the embryonic induction in detail. Dear students, up till now we have studied the concept of differentiation, and I hope that it is now clear to all of you that how the cells, they impact each other for growing into specific organs and tissues depending upon the functions. And it was also discussed earlier in the previous lectures that the cells, they decide to become the part of a certain organ depending upon their interaction with the neighboring cells as well. Means, in addition to the cytoplasmic control, there is also the effect of interaction among the neighboring cells. So, embryonic induction means the capacity of some cells that are present in the embryo, they have the capacity or ability to invoke or start the development of the other cells. Means, they push or they force the other cells to become certain organs or form certain organs. Such capacity and such process is known as embryonic induction. This whole phenomena is taking place within the embryonic cell, and one layer of cells, it affects the other layer to form the specific organ or organ system. For example, in the previous experiment we have seen that mesodermal cells, when they were removed, the ectoderm was unable to form the nervous system. Although in the process of neurulation during chick development, we have seen that notochord and neurulation process was completed by ectoderm, and it was the ectoderm that actually formed the neural plate, and it folded inside to form the neural tube. But in the experiments that are performed by Spearman on the frog's embryo, it was revealed that it is not only the ectoderm that is controlling the formation of the notochord and ultimately the nervous system. Instead, the mesodermal cells that are present between the ectoderm and the endoderm, the mesodermal cells are also controlling the formation of nervous system. And if mesoderm is removed by any means, the ectoderm will not be able to form the nervous system. So, this impact of mesoderm on ectoderm for the development of nervous system is a very good example of embryonic induction. This phenomena was studied by Spearman as well as another scientist, Mangold, in the year 1924. They also give the experimental evidence for this phenomena. For this experiment, they took two salamander embryos, number 1 and number 2, and both these embryos were at gastrulation stage. At gastrulation stage, when epiblast and hypoblast, they are differentiating into the three layers, ectoderm, mesoderm and endoderm. So at this stage they took the embryos, and from gastrula number 1 they removed the dorsal blastopore lip, which is actually nothing else but the mesoderm. So they removed this mesoderm, they cut a 180-degree segment and grafted or pasted this segment into the second gastrula. This second gastrula has now two kind of the tissues, one its own and the another one the grafted one. So they removed this dorsal blastopore lip and grafted it at the ventral position of the gastrula number 2. Now what they observed was that there was development of notochord and somites. I hope you are well aware of the somites. They are specific group of cells that give rise to different tissues, muscles and other organs in the future. So in addition to its own notochord and somites, the external grafted mesoderm gave rise to somites and notochord in the gastrula number 2. So it will have two types of notochord, one its own primary and the second one the secondary. This primary dorsal blastopore lip of this gastrula, which was its own, this induces the development of the grafted tissue. This means that the already present layers of the cells in the gastrula number 2, they induced the development of the newly arrived or the grafted dorsal blastopore lip, and ultimately that also changed into the notochord and somites along with its own nervous system. Now the embryo will have two nervous systems, one the primary its own and the other one secondary that was formed by the grafted tissue. So as the primary cells they induce the development in the secondary cells, so it will be known as primary inducer because it is inducing the development in the other one, and this phenomena will be known as primary induction. So from here it becomes crystal clear that the development is completed by the mutual interaction of all the cells present in the embryo. Although all the cells have the capability to form a complete organism, yet they interact in a systemic way so that no abnormality occurs, and ultimately the formation of organs is completed in an efficient manner. I hope the topic is now clear to you. In the next lecture, we will discuss the process of aging. Till then, take care of yourself. I hope the lecture is now very much clear to you. Keep watching office.com.pk and keep coming for more easy and handy lectures. Thank you so much for your time. Allah Hafiz.